record on this computer. It says it's recording, so I suppose here we go. I'll share the screen that I'm working on. Pull. Okay, I think this is working. Everyone's still here. You guys can see this. Okay, so in one of the lab sections, someone asked for like an example of a manuscript, basically, and I have one good example from last fall's semester. So I didn't teach last semester, so like I don't have a pool of these to pull from. Dr. Small shared this one with me with permission from the group. In order to share this with you guys, I would need permission from the group as well. So I just have to reach out to them. But I'm hesitant to do that because I want these manuscripts to be like your own works. And I know some students feel like they have no idea what they're doing. But like, so this example that, like I said, scored really well last semester. I mean, it literally just looks like the manuscripts you guys are reading. The primary research articles that you guys are supposed to be reading and citing and stuff, it's set up just like that. So, you know, like this must be materials and methods. Scroll up to section 2. Yeah, so like, you know, you would see this in almost any primary research article you go through. Section 2 is materials and methods. So like 2.1 is husbandry and exposure. So like this is our treatment paradigm. 2.3, well, let's do 2.2. So they did their assays in a slightly different order last semester. So last semester they started with morphology. We're ending with morphology, but because they did morphology first, their first real experimental paradigm section is morphology, right? Then they did protein quant, which is the amino blot. Then they did the section we're doing right now, reverse transcriptase PCR. So like they literally have a subsection in their materials methods section for each one of these. And it's as far as organization goes, it's as easy as just going 2.1, 2.2, 2.3, 2.4, you know, in whatever you want order you want. Here's an example of like how you can report your primer information. A lot of publications just do this now. They just stick in a table because it's a lot more organized than like getting into this section of text and just like slapping together AGC, ATT. It's a lot more organized and nicer to look at if it's in a table like this. So that's a loose example of how you can set up your manuscript. And then like section 3, I scrolled past it, is results section. And their results section is broken up similar to their methods, right? So 3.3 is their report of the results from reverse transcriptase PCR. Section 3.4 is their report of results from that, and so on and so forth. And then section 4 is discussion. You guys all set with that? I don't even think any of you three asked, but somebody asked. So if anybody's going to take the time to watch this, I said it and I gave an example. But so what we're really here for today is to I'm going to show you guys how to like quickly analyze your reverse transcriptase PCR data. So this is an example from this manuscript. So you guys are going to want to mark up your data similar to this, right? So everybody has 2 images of, well, everybody except for one group has 2 images of 2 gels. So over here on the left, just like you guys are going to see, is your DNA ladder. You can use the vendor information image that I uploaded to each section's Brightspace page, and you're going to want to like highlight which what size each of these DNA ladder chunks are, right? So I think I mentioned this in all sections. You know, this really faint one down here is 250, 500. They've labeled it 500. The first bright one is 1000, and they've labeled it as 1000. So this will make it a lot easier to scroll across and, you know, estimate the size of your bands. So, you know, like these bands right here, it looks like this is TBP, so everyone's going to have this on their gels. TBP is just shy of 500, right? And then, so this one over here, it looks like they did PDX1. lines up with the 1,000 kilobase pair chunk in the DNA ladder. So we know these DNA fragments are 1,000 base pairs. So long-winded explanation, you're going to want to mark them up like this because you're going to want to tell me what each band is. So if you followed the organization structure I had on the PowerPoint slides, you would have organized it just like this, right? So this is, they did TBP first. So this is gene 1, no cDNA. And then there are 3 other treatments. They're calling this no DNA. So no cDNA control, right? That shouldn't have any signal. And theirs don't. And then this one's just your control treatment. And then DMSO. So these guys use phthalates, and then your drug. So you want to annotate it just like this, so I know what band is what. Any questions about that from the 3 spectators? And it looks like, I don't know how to do this. It might be something you can do with a tablet. It kind of looks like they opened this image and edited it themselves, like with a stylus. You can also do this by throwing it right in a PowerPoint, and you can like enter a text box over it, type these things up. You can do all this in PowerPoint. It's how, like, if you guys have seen some of the videos I've made with animations, it's how I would do it. Okay, so annotate your gels. And then, so this is what we're ultimately trying to make. We're trying to make a bar graph that compares gene expression across different treatment groups within each different gene. So like this is neuro D1, with my graph, let's just call that gene 1. And then they had multiple bands. So some of you guys have multiple bands. Feel free to analyze those for the sake of analyzing them, but I won't hold it against you if you only analyze the band that's the band of the product that's where it's supposed to be. So like if you have a band, if your gene product's supposed to be 500 and you have a band at 500 and a band at 250, you can analyze both if you want. It's fine if you don't, just analyze the band at 500. So gene 1, gene 2, gene 3, gene 4, right? And then because these are all ratios, the y-axis over here, they've labeled it as control pixel to pixel ratio. You can also label it as gene expression normalized to TBP, because in reality, these the numbers we're graphing here are just normalized gene ratio. They're gene ratios normalized to TBP, which is our housekeeper gene, and everything's in ratios. So there aren't technically units on it. So like even over here, it says pixel to pixel ratio. If you were looking at this as units, the units would disappear. So you can label it as either or. Just make sure you label it. Make sure you label it and make sure you label it accurately. Don't put like Snuffleupagus from Sesame Street over here. That's not a correct label. And okay, so for the groups that had phthalate treatment, you have to do 2 normalizations. So you normalize to control when you graph. So like DMSO to control, and then treatment to control. So that's one set of ratios you're going to graph. And then you also have to normalize your treatment to DMSO. And I'm going to show you how to do that in this. The groups that just did ethanol, you basically just have ethanol to control treatment 1, and then ethanol to control treatment 2. Any questions? No. Okay. All right. So this is basically what we're going to try and create. I've already gone through and made it. And like I said, if you are good with Excel, you can do this in about 5 minutes. But I'm going to try and go a little slower and show you guys some of the tricks to make Excel work better for you as well. And I pulled this from a group on Tuesday. Oh, right there, group 5 on Tuesday AM. So it's the data I'm going to graph is as if the animals were treated with phthalates, not ethanol. But like I said, that the ethanol treatment will just have like drug treatment 1 versus control, and then drug treatment 2 versus control. All set? Okay. All right. So this is what I sent everybody. If you remember from my PowerPoint in class, I had like those mock blue rectangles over the bands in our gels. So this is basically what, after you tell the computer to analyze the pixel intensity in each one of those rectangles, it spits out a lot more information than this, but I trimmed it down so you guys don't have to see it. But this is basically what you guys should see. And where is my preview? There it is. So these numbers are from this gel, right? So just like I showed in the PowerPoint, each one of these is going to have its own blue rectangle, which the computer is going to register as a lane. So like this is lane 1, lane 2, lane 3, lane 4, lane 5, etc. So you'll notice over here on the volume intensity output information that you guys get, if there's no signal in a lane, the lane's completely blank. Like basically the software read it and was like, there's no signal here, so you get no information. So it's just 0. But otherwise, like I mentioned this in all the classes, the value that's output here is representative of how bright each of these bands is. So like lane 2 here, we have a value of 480,000, right? So this band. Whereas lane 3, we have a value of basically 746,000. So this one's roughly, this band has roughly twice the amount of basically light as this one. Does that make sense? I think I mentioned this in each of the presentations, like my mock numbers weren't exact because I made them up just to walk you guys through the process, but the data you actually get from your gels is representative of this stuff. And so what is this, 4, 8, 10? So like over here, lane 10 is 250. Like it's saying this is basically about half as bright as this guy. So the numbers correlate with the brightness of the bands. And then for basically everyone except the one group who only has one set of wells, you guys have this restart over here where it's going to say like lane 1, lane 2, lane 3, 4 after lane 12. That's the data from down here. Okay. Okay. So let's start analyzing. So we know that we have 4 treatment groups, right? So, well, let's be complete. No cDNA treatment. Okay. 1, 2, 3, 4. Merge. No cDNA. Control. Since this guy is phthalate, we'll do this, and then I'm just going to call this drug. Yeah, actually, I'm going to make that a little bigger. Does that help? Okay, so we know there's 4 treatments, right? And then genes generically, and this is going to depend on how many genes you guys all had, but this group has 4. So we're going to do 4 genes, right? And I'm just going to write gene 1, gene 2. If you guys didn't notice about Excel, Excel tries to be very helpful. So it's going to detect this pattern, hopefully. Gene 1, gene 2, you can select those, grab this bottom right corner, pull down, and it should auto-populate gene 3, gene 4. And I say Excel tries to be helpful because like sometimes that works, and then other times Excel will say, oh, you want this box to be gene 1 and this box to be gene 2. So like inappropriately copies the pattern it's trying to recognize. But it'll work great here. Okay, everyone with me so far? Cool. All right, so these guys laid their genes out the way I basically instructed everybody to. Again, on the PowerPoint slide, I said like 4 lanes, gene 1, 4 lanes, gene 2, 4 lanes, gene 3, etc. And they kept everything nice and labeled, so it looks like everything is the same, right? They don't have, in this set of genes, they don't have a band up here and a band down here and then a band over here, making things look inconsistent. So we're confident that these 4 lanes are gene 1, these 4 lanes are gene 2, these 4 lanes are gene 3, and then these 4 lanes are TBP. So if you did everything, or if everything worked out the way it was supposed to, this next part is really easy, which was the point and the design and the thought process behind this. So you can literally highlight the volume intensity from those 4, copy, paste, right? That's gene 1, copy, pasted, done. Gene 2, copy, pasted, done. Gene 3, copy, pasted, done. And so you'll notice that for whatever reason, there's no signal in this lane. That could be because, I mean, it could be because of so many things. Like I could spend 30 minutes coming up with why it might be blank. It's not important right now. It is blank. And then TBP, copy, paste. I'm going to change this to TBP. Okay, so now we have a nice, neat organization of the pixel intensity for all of our samples and all of our genes, right? And the first thing we want to do is normalize our signal of each gene to TBP. So like over here, I'm basically redoing all this, but so this top section is raw data, and then we're going to put a section right below that that says normalize to TBP. So gene normalize to TBP. Okay, I'll do 1. You could technically just do this, it's faster. I'll do L4 to show you guys what that looks like. And how did I do it before? I left those out. Okay, so because these are still in line, these are still in line, like this is the same treatment. So this column is no cDNA, this column is control, this column is DMSO, etc. So no cDNA has no signal, like it's not supposed to. I mean, it's not supposed to have signal, so those are supposed to be blank. So we're just going to leave those alone. All right. So the first step is normalizing gene 1 expression in our control treatment to TBP. So we want to go our value for control gene 1 divided by control TBP. And one of the cheats you can use with Excel, if you put a $sign in front of the column and row information here, it's going to tell Excel to lock that cell in place. So I hit enter. It does the math for me. 480,000 divided by 718,000 is about 0.666, right? So I don't know whether to make the assumption that you guys know how to do this or not, but so if you don't know, if you guys don't know, you can make Excel copy things really quickly, like copy formulas very quickly by also doing this. So I can just grab the bottom right corner of that cell, drag down, and it's going to copy that formula. So it copied the formula. But like the way Excel works is it copies with respect to where the cell identity is. So it copied the formula, but because it's in one cell lower than where it was before, it's going to shift the cells it's using in that formula down 1, right? So instead of looking at control gene 1 information now, it's looking at control gene 2 information. Let me show you guys what happens if you don't lock it. Okay, so I just typed same formula, this divided by this, but I didn't put the $signs in. So if I drag and drag, now I have all these divided by zeros. So again, the original box, this divided by this, the one box below it, like I said, it's shifted, it's basically shifted both cells down one now, because by copying it, I'm telling Excel, use the same formula, but shift down. And then like this one is 2 down. This one's, oops, edit and do. And then this one's 3 down. Okay, so that's why you use the dollar symbols, because the dollar symbols lock that cell in place. Right, so all of these, this stays the same. And then TBP, if you normalize it to itself, is 1, right? 718,000 divided by 718,000 is 1. Any questions about that? Cool. All right, so now we're going to copy the formula I just used, paste it over here, and like, so that's what we're going to do to make this a little faster. But now if we click on this, remember I put those dollar signs in, so it locked it in place when I copy and pasted. You can just drag it over. Right now, now with the dollar signs in place, it's saying it's going to lock onto this cell. This one's still over. Oh God. Oh God. Edit undo. Okay, I just messed it up, but I edit undo and it's better. So this one is over here. Cell's still locked, so I'm dragging it over. Enter. Right. So now I have those two set up. I can select both like that, drag it both down, and ta-da, it did all that math for me automatically. Any questions? Nope. Okay. Okay, so that was step 1 of normalization. Now the numbers we have are basically each of our genes normalized to our housekeeper gene TBP. So like I said in class, now we've compared the expression, basically the quantity of expression of gene 1 to the quantity of expression of TBP. So what that basically is saying is controls. So the value of this pixel intensity divided by the value of this pixel intensity. And that ratio comes out to be 0.66. So like this band is 2/3 as bright as this band, which translates to there's 2/3 the amount of this DNA as there is this much DNA. And then if you go like, treatment by treatment, this guy is the same. So this is about 1/3 of this. And with our drug, this is technically a little brighter than this, is what that's saying. And so why that's important is, so like between these two groups, you can see that, you can see it in the numbers. So like this band has a value of 718,000 and this band has a value of 120, or is it one? That's 1 million. Yeah, 1,120,000, right? Like this one's way brighter than this. So what that means is in total, in this sample, there's just more DNA. So if there's in total, if there's more DNA floating around, you would expect this band to be brighter regardless of treatment, potentially. Does that make sense? Okay, so that's why you have to normalize within each control. That's why you normalize your gene of interest signal to your housekeeper gene. So that's what all these are. Okay, so now we're gonna so that's good. You could technically graph this if you wanted to, but we want to know how our gene expression is changing compared to our control group, right? The group that has had nothing done to them. So the way this is going to look when we're done is we're going to have a normalized to control section and then a normalized to our vehicle section. Again, these guys used phthalates. So another extra comparison they can do is normalizing drug 1 to DMSO. Okay, so let's do that. So this is DMSO to control. And this is drug to control. And this is drug to DMSO. Right. So equals DMSO divided by control. And since I want this to, I'm going to lock it in. You don't even have to because it's probably faster not to, but it's faster not to. I lied. Okay, so this is still DMSO divided by control. Okay. Now we want drug to control. So I'm copy pasting. We want our drug signal divided by our control signal. So we just move this over. And then this one's probably already be done for me. Oh, it would have. All right. Okay. So drug divided by DMSO. Everyone's still good? Yep.
So for our group, we didn't have anything in the control. So if we were to do the drug divided by the control, we would get an undefined number. So what do you want us to divide it by?
What can we do about it? Can you shoot me an email what I can try and do?
Yeah, I'll just email you.
Yeah, can you shoot me an email with the gene that you're missing?
Okay.
And if I'm if we're all lucky, someone else may have run that gene. And maybe I can give you that data. I mean, the alternative is basically rerunning these. So like anyone who had stuff not show up like it was supposed to, the real solution, like if this was a real lab situation, is you would rerun the whole thing, which people can if they want to. You would just have to do it like in the background of what else we're doing every week. Does that make sense? I mean, if it's only one gene, it's not like the end of the world. You should still have other genes to compare, other genes to report data for.
It was all of our genes, so.
Oh, none of your control?
Redo it, yeah.
Oh boy.
It was all of them, yeah.
Are you in Wednesday's lab, Ashley?
Yes, I am.
Yeah, that doesn't really change anything if the control's gone. I mean, well, if it was phthalates, you could at least normalize to DMSO. I mean, technically you should.
Okay.
Technically you should. I'm not going to make you. You can still technically report the ratio to TBP.
Okay.
That'll, that says something. If the ratio changes, yeah, I mean, if the ratio changes, technically, you could compare the way different. No, you don't have control.
We'll just redo it. I'll email you.
Okay. All right. Okay, so drug normalized to DMSO. So that's just drug value divided by DMSO value. And now, because I didn't put those dollar signs in, I can select all three and I can just drag them down. Boop. So now we have DMSO. Oh, what did I do? You're just supposed to hover. DMSO normalized to control for gene 2. DMSO normalized to control for gene 3, etc. So this one isn't working because there's no value for the drug there, right? Okay. Any questions about that? No. Okay. And to help Excel do its job, you can plug this in. And so then you should be able to select all this. No, something in the way. There we go. Insert. Excel. My Excel was helpful when I did this. So I got away with just going to Excel, recommended charts, insert recommended charts, and then the first recommendation is the type of graph we want. So now we have this. And because like these are gene 1, gene 2, gene 3, but like the blue bar series is our DMSO normalized to control. The orange is our drug normalized to control, and then gray is our drug normalized to DMSO. And then because we don't have drug data for gene 3, it's blank. So technically this x-axis would be complete because it has our gene information and we have our legend of what each one of these is. Your chart title, these guys used mRNA expression of gene target. I titled my other one effects of phthalates on mRNA transcription because that's what we're looking at. I'll just type that in, effects of phthalates. And so if you're typing this, you're going to want to report, I don't remember the doses off the top of my head. Whatever it is, MIG per mil phthalates on mRNA expression. All right, so just make sure your graph has a title. So then, right, gene 1, gene 2, gene 3, the treatment groups. The one thing this is missing so far is a y-axis title. So if you select y-axis, I don't even think you need to select y-axis. You can just select the graph. You go up here to your top left, add chart element, axis titles, primary vertical, and then you can type in whatever you want that to be. And what did I say? Gene expression wise to TDP. Okay. Why'd you move? So now what we're looking at, sec. Okay. So now what we're looking at is the expression change in each of your genes, right? So DMSO compared to control for gene 1 didn't change very much. So that means DMSO had no effect, relatively no effect on the expression of gene 1. Does that make sense? So then our drug compared to control, so our drug had an effect. Our drug basically increased gene expression twofold, right? That's what this is saying, twofold. And that because our DMSO didn't change our gene expression, our drug to DMSO ratio is the same. So like we want these to be about the same because that means DMSO had no effect. And then so over here, this is basically saying the DMSO had a slight effect on the expression of gene 2 compared to control by decreasing it, right? So it's almost, the expression of gene 2 is almost half in the DMSO treated group that is the control. Does that make sense? And so this is actually a good example of vehicle effects. But if you normalize drug signal to control signal, it's about 1, which would mean the drug treated group has about the same expression profile as the control treated group. Does that make sense? Which is why you need to do the additional normalization where you compare the expression profile in your drug treated group to your DMSO treated group. So like here, like I said, this basically shows that DMSO decreases expression of your gene. So it makes it look like it's half. So when you normalize your drug signal to your DMSO, which is the vehicle, you can actually see that technically your drug increases your expression of your gene by like twofold compared to DMSO. And that would get washed out, right? That looks like it's washed out if you weren't comparing your drug to your vehicle. And then gene 3 is empty. And so like, that's it. That's how you make the figure. You know, when you write your figure legends, again, you can model your figure legends off of the information in figure legends from primary research articles that you guys are reading. And you just want to, you know, make sure you have specific information in here about the contents of the figure. Any questions? Cool. All right. So that's pretty much it for this. This was like I said, a relatively quick one. If things were organized the right way, it's like, if you're good with Excel, this only takes 5 minutes to put together. If you have to reorganize some of your lanes, this might take a little longer. But if everything was done the way I had instructed it / set up, and when I say instructed, it's basically like the plan the whole time was to do it the way I had it set up, so that when we get to this analysis portion, it's really easy. Okay. So if no questions, I guess that's it. And then I'll see you guys. I'll stop sharing my screen, and then I'll see some of you guys on Monday, and then the rest on whatever day, Tuesday, Wednesday, or Thursday. Cool. All right.
Sounds cool.
Awesome. Thank
That
you.
wasn't too
Thank
bad.
you.
Yeah.
Thank you.
Yep. See you guys. Have a nice
Have a good
weekend.
one. You too.
Bye.
I stop recording.